Okay, so we are thinking about Chargaff's rule. So Chargaff, okay, he did a bunch of research and he figured out in organisms the amount of A is equal to the amount of T, and the amount of G is equal to the amount of C in organisms. So we're talking about DNA. So in any organism, those amounts are equal. And the other thing that we know is that all of those together equal 100% of the DNA. Okay, so A plus T plus G plus C is going to be 100. So we can use those two to figure out how much of each nitrogen base is in each organism. So here we have some examples. I have a fruit fly and a bear. So we're going to start with the fruit fly. And here we're given that the fruit fly has 28% adenine, or A. We go to Chargaff's rule, we know that the amount of A, and this one I have them next to each other, but always check, A is going to be equal to the amount of T. So then we know, okay, then the fruit fly must have 28% T. So given that, then we have to figure out how much guanine and cytosine are in this organism. So again, we know A plus T plus G plus C is 100. It's 100% of the DNA is made up of those. So what we then need to do is just take what we have. So we have A and T, so we add those together. So A plus T, so 28 plus 28. You just get out your calculator and you add those together and you get 56. Okay, so we know A and T equal 56% of the fruit fly's DNA for their nitrogen bases. Okay, so now we need to figure out, okay, so then how much is G and C? So we take our 100% of the DNA, we subtract out the A and T, 56%. Okay, so you just take 100 minus 56, and you're left over with 44%. Okay, so we figured out A and T. We have those, okay, and we're left over with 44%. So we have this G and C. We know those together are 44% of our DNA. We go back to Chargaff's rule. We know G has to equal C, so they're going to be the same. So all we do is we're like, well, they have to be equal, so we'll just take that and divide it by 2. We take 44 divided by 2, and then we know, okay, 22% is G and 22% is C. Okay, let's do this again. We can do it with the bear. We're going to do the same sort of thing. So let me erase this stuff a moment. And then we'll do it for the bear as well. Okay, so for the bear, we are given something different. Let's do it in blue. Okay, so for the bear, we're given the G. So if we know G, we know C. So we fill that one in. So we have G and C. We want to figure out A and T. So we know 100% of the DNA is made up of all of them together. We're going to subtract out the G and C. So add those two together, you get 22. Okay, so then we figure out how much is left. What percentage of the DNA is left? So we have 78% left over. So we know A and T together make up 78%. So we just divide that by 2. And we get 39. Okay, so as long as you know Chargaff's rule, then you can figure out 2 of the pairs. Then you just subtract what you have from 100. Whatever you have left, you divide by 2, and that's going to be the other 2 nitrogen bases. Okay, so that's Chargaff's rule. The other thing we need to know is about DNA structure. So DNA, we think of it as a ladder. Okay, ladder has 2 sides and then rungs in the middle. So that's like the base structure of DNA. How we get this ladder is we build it out of something called a nucleotide. So a nucleotide is made up of a phosphate attached to a sugar, which is attached to our nitrogen base. Okay, so we've got our phosphate, our sugar, and then our nitrogen base. Okay, again, those nitrogen bases can be the A, T, G, or C, depending on which one's attached to it. So basically with DNA, we take a bunch of these things, the phosphate, the sugar, and the nitrogen base. These three things together are called a nucleotide. So in DNA, all we're doing is we're taking nucleotides and putting them together. So we string a bunch of them together and we come up with this ladder. So let me show you in our class stuff a moment. Okay, so if we go in here and we look at our DNA, let's look at this one. Okay, so here we can see, again, we have this we have the phosphate, which is the orange circle. We have the sugar, which is this blue, and then we have our nitrogen base. And you can see that they get connected together. So we have those nucleotides that are attached together, and that makes up our rung, our ladder. So the sides of the ladder are this sugar and phosphate. The middle of the ladder, the rungs, are the nitrogen bases stuck together. And then the other side again is that sugar and phosphate. Notice too here we've got, if you look at this little dot, kind of like a nose, the direction of it we can see. This side has the nose pointed one direction, see the arrow. The other side has the nose pointed the other direction. They're just opposite of each other. It's like a highway. One strand's going north, the other strand's pointed south. And that's just, it ends up being an important structural thing in DNA, but we call it the anti-parallel structure of DNA. Again, if we look at this sugar, you know, the point goes one direction here and the other here. Okay, and again, these are nucleotides that we put together to build that DNA up.